National 5 biologists. Let's do this past paper multiple choice questions together. So number 4 says lipase is an enzyme that speeds up the breakdown of fats, producing an acid that decreases the pH. 3 test tubes were set up to investigate the action of lipase on fat in milk. Okay, so let's get a couple of things clear before we move on here. So lipase is our enzyme. Okay, that's well worth remembering. And we've got the milk, which is going to be our substrate. Okay, and it says that it produces an acid. That is going to be our product. Okay, so it's good to get all these terms out of the way straight away. Okay, so it says the pH of each test tube was recorded at the start of the experiment and then again 20 minutes later. In which of the test tubes would the pH stay the same? Okay, so let's have a look at the options. So first of all, we've got R. Okay, now in R, we've got the substrate in the milk. Okay, the substrate is the fat, which is in the milk. I'll just write that down just so we're clear on that. That's the fat in the milk. We've got the enzyme, which is the lipase. So when we have a substrate and an enzyme, a reaction is going to take place. And what do we get at the end of a reaction? Is we get a product, and our product here is acid. So therefore, in which of the test tubes does the pH stay the same? Well, it's not going to be R, because R is going to become acidic. It's producing the product, so therefore it'll become more acidic, okay? So we know it's not going to be R. Okay, the next option is T only, so let's have a look at T. Now the issue with T is that we have boiled lipase. Now when you boil an enzyme, it will become denatured. Okay, and if it becomes denatured, will a reaction take place? We'll know. So therefore, is T going to stay the same pH? It probably is. Okay, so T is going to stay the same pH. Okay, we can eliminate R, A, and C there because they both involve R. But we need to just see if S is going to work because that's an option for us in D there. So we've got milk and water this time, okay? So this time we are lacking the enzyme because we've replaced it with water. That would be our control experiment. And because there's water there and no enzyme, there'll be no reaction, which means no acid being produced, and therefore the pH will stay the same, okay? So the answer we're looking for there is going to be D. Okay, for number 5, a 100-gram sample of hemoglobin was found to contain 15 grams of leucine, okay? So 15 grams of leucine from 100 grams of hemoglobin. What mass of leucine would be found in a 5-gram sample of hemoglobin? Okay, so what it's saying is when there's 100 hemoglobin, that would have 15 grams of leucine. And this time when the mass is 5 for hemoglobin, what would be the leucine? Okay, so we're just going to simplify both of these by a similar factor. So how do we get from 100 down to 5? Well, we divide that by 20. Okay, 100 divided by 20 gives us 5. And so this time we're just going to do 15 divided by 20. And that should give us our answer, okay? 15 divided by 20 is going to give us 0.75. Okay, so what we're doing here to get from 100, where we have 15 leucine, to 5 grams, where we have 0.75 leucine, as we divide both sides by 20. Okay, so we're basically using a similar common denominator in both of the sides of that ratio there. Okay. Question 6. A culture of 350 bacterial cells was genetically engineered to make a human protein. Only 210 of those cells were successful. Okay, so only 210 out of 350. Okay, and because we write our working like this, we've already done half of what we have to calculate. Okay, and because it wants a percentage, we do the successful divided by the total, and we times that by 100. Okay, and 210 divided by 350 times 100 is going to give us 60. Okay, so we're looking for 60 as our answer for that particular one. Okay folks, I hope these helped. I'll see you in the next one.